natin ang tungkol sa GMO.
Ano pang hinihintay nyo? Ayan na!
Hi guys! Nandito ako ngayon para talakayin ang timeline of genetic modification in modern agriculture. For thousands of years, people have worked to improve crops, livestock, and the food that we eat. In the 20th century, ang mga scientists ay nakakita ng paraan para i-modify ang mga pagkain in precisely na sa paraang babaguhin nila ang DNA ng isang organism. Ang prosesong ito ay tinatawag na genetic engineering that produces genetically modified organism or GMOs. And I'm here to tell you about the timeline of genetic engineering or genetically modified organism. 40, ang mga plant breeders ay natutunang gamitin ang radiation or chemicals to randomly change an organism's DNA. In 1953 naman ay na-discover ng isang chemist na si Rosalind Franklin at scientist na si James Watson and Francis Crick ay na-identify ang structure ng DNA. Noong 1973 naman, ang biochemist naman na sina Herbert Boyer at Stanley Cohen ay nag-develop ng isang genetic engineering na sa paraang ini-insert nila ang DNA ng isang bakterya sa isa pang bakterya. In the 90s, the first wave of GMO products created through genetic engineering become available to consumers, like summer squash, soybeans, cotton, corn, papayas, tomatoes, potatoes, and canola. Not all are available for sale. In 2015, FDA approves an application for the first genetic modification in an animal for use as food, a genetically engineered salmon. In 2016, Congress passes a law requiring labeling for some foods produced through genetic engineering and uses the term bioengineered, which will start to appear on some foods. In 2019, FDA completes consultation on first food from a genome-edited plant.
Paano nga ba nagawa ang GMO? Genetically modified organisms include bacteria and yeast, insects, plants, animals, and mammals. The term GMO is also known as living modified organism. Ano nga ba ang genetic engineering? Ang genetic engineering, or also called genetic modification, ay isang proseso na ginagamit ng laboratory-based technologies para mabago ang DNA ng isang organism. So how are GMOs made? First, ang mga scientists ay mahanap ng isang desired trait of plants, animals, or even a bacteria. When they found one, scientists copy the genetic code that contains that trait and insert into the DNA of the crop they want to improve. Napapaisip pa rin siguro kayo kung ano ang mga karaniwang ginagamit sa paraan na ito. Here it is. First is the microinjection. It is a physical and direct transformation technique which uses a fine chip glass needle to deliver DNA into the cell. Karamihan, ginagamit ito sa mga animal cells. Ginagamit ito sa pamamagitan ng injection. Second, the electroporation. It is a physical and direct transformation technique which uses an electric field to deliver DNA into the cells. It is mostly used in plant cells. Ginagamit ito sa pamamagitan ng pulsed electric charges. Ang mga napiling genetic material ay nilalagay into the seeds or bagong fertilized eggs of animals and it will grow within the new genes. Inoobserbahan nila if it adopts the desired trait. They also observe and making sure that their GMO they made are safe to grow, safe to eat, and safe for the environment. After how many years of research and testing, in-approve ito ng government that can be planted by farmers.
Has genetic engineering changed plant and animal breeding? Ginagamit ang lahat ng mga traditional na paraan ng pagbabago tulad ng selective breeding at crossbreeding upang maparamin ang mga halaman at hayop na may kanais-nais na mga katangian. In the United States, ang field na ito ay defined by the Engineering Council for Professional Development bilang creative application of scientific principles to design or develop structure, machine, or manufacturing processes. Related with engineering is a great body of special knowledge. Isa itong preparation for professional practice na involve ang mahirap na training para sa application ng knowledge patungkol dito. Sinasabi na ang standard of engineering principle o professional society, karamihan dito ay organizing national or regional basis. Kasama ang lahat ng members, acknowledging the responsibility to the public over and above responsibilities to their employer or to other member of their society.
Ano ba ang role ng GMOs sa ating environment? Ang management of environmental issues ay another area na kung saan ang GMOs ay maaaring gamitin. For instance, ang ilang mga bakterya na maaaring makabuo ng mga biodegradable polymers at ang kakayahan na i-transfer ang mga capabilities nito to microbes na simple sa culture in the lab may enable the widespread draining of plastic industry. May isang isang negosyong British na tinatawag na Zeneca ang lumikha ng Biopol, isang biodegradable na plastic na ginawa ng mga microorganism. Noong 1990s, ang Ralstonia eutropa is isang GM bacterium. Ito ay ginamit upang likhain ang plastic sa pamamagitan ng pag-convert ng glucose at isang bilang ng mga organikong acid sa isang flexible polymer. Ang mga GMO na binago sa genetics upang magkaroon ng kakayahang tumunaw ng mabibigat na metal at langis ay maaaring maging isang pamamaraan ng bioremediation. Despite the fact na maraming potential advantages for society ang GMOs, mayroon pa ring ilang risks na involved lalo na sa industry ng pagkain. Ang mga risk na maaaring idulot ng mga pananim na GM sa kalusugan ng tao ay malawakang pinagtatalunan. Ang mga allergenic na katangian ng mga pananim, halimbawa but in contrast, in the United States, foods containing GM ingredients didn't need specific labeling. The unidentified risks to food safety were a major point of contention for many opponents of GM products. But despite the worries of some consumer and health advocacy groups, particularly in Europe, numerous scientific panels, including the US, according to the Food and Drug Administration, eating GM food was safe, even when the product contained genetic material from extremely unrelated creatures.
A GMO plant step by step. The following example gives a general idea of the steps it takes to create a GMO plant. This example uses a type of insect-resistant corn called Bt corn. Keep in mind that the process for creating a GMO plant, animal, or microorganism may be different.
So first step is to identify. To produce a GMO plant, scientists first identify what trait they want that plant to have, such as resistant to drought, herbicides, or insects. Then, pag nakahanap sila ng isang organismo sa halaman, hayop, or microorganism na mayroon ng katangiang iyon sa loob ng mga gene na ito. So, second step is copy. After mahanap ng scientists yung gene with the desired trait, makopyahin nila yung gene. For Bt corn, they copied the gene in Bt that will provide the insect resistance trait. Third step is insert. Para ma-insert yung gene into the DNA of the plant, yung scientists gumagamit ng tool. By inserting the Bt gene into the DNA of the corn plant, scientists gave it the insect resistance trait. This new trait does not change the other existing traits. Lastly is grow. In the laboratory, scientists grow the new corn plant to ensure it has adapted the desired trait. If successful, scientists first grow and monitor the new corn plant, now called Bt corn, because it contains a gene from Bacillus genesis in greenhouses and then in a small field test before moving it into the larger field test. GMO plants go through in-depth review and tests before they are ready to be sold to farmers. The entire process of bringing a GMO plant to the marketplace takes several years.
Hanggang sa dulo ng mundo, hanggang maubos ang ako, hanggang gumulong ang luha, hanggang mahulog ang tala. Masdan mo aking mata, di mo ba nakikita? Ako ngayon lumipas at nasa langit na. Gusto mo ba sumama?